For over a century, Egypt kept one secret that no pyramid, no tomb painting, and no golden mask could give up. Somewhere inside every mummified pharaoh, every noble, every priest ever pulled from the sand, the one thing modern science actually needed most had already been destroyed, quietly, chemically, centuries before anyone with a microscope arrived to look for it. In 1902, archaeologists excavating a hillside cemetery in the Egyptian village of Nuwayrat, south of Cairo, pulled a skeleton out of the ground still sealed inside a large ceramic pot. There had been no elaborate mummification, no golden mask, no inscribed sarcophagus, just bones folded inside plain-fired clay, tucked into a rock-cut tomb chamber in the cliffs above the Nile. The remains were shipped to Liverpool, added to a growing university collection of Egyptian antiquities, and then survived something almost none of the pharaoh's own tombs had to survive: a direct hit during a German bombing raid in the Second World War, which destroyed the majority of the museum's human remains collection around it. This particular skeleton, tucked safely away in storage, happened to escape. It then sat largely unexamined for the better part of a century. Nobody realized what they were actually holding until 2025, when a team of geneticists finally managed to do something that had defeated every previous attempt in the entire history of Egyptology: extract a complete, usable genome from a body that had been dead for close to 5,000 years, older than the Great Pyramid of Giza itself. For decades, ancient Egypt was considered close to a genetic dead zone, a strange blind spot in a field that had otherwise mapped the deep genetic history of Europe, Asia, and the Americas in remarkable detail. The same intense heat that preserved so much of Egyptian civilization in stone, paint, and papyrus was actively destroying the one kind of evidence modern science needed most: DNA, which breaks down rapidly in hot climates, and even faster inside bodies that had been deliberately mummified using chemical preservation processes that, geneticists eventually discovered, contaminate and degrade genetic material even further than exposure to heat alone. For over a century, that meant the deepest, most fundamental question about the people who built the pyramids, where their ancestors actually came from at the genetic level, before any of the written record even began, remained functionally unanswerable, no matter how many mummies archaeologists uncovered. Then one unmummified skeleton, buried by accident of circumstance in exactly the right conditions to survive intact, finally broke that century-long silence. And what his DNA revealed wasn't just older than anything recovered before. It rewrote the assumed timeline for one of the most significant population movements in the ancient world, using nothing more than 2 teeth and a burial pot that had spent over 100 years sitting quietly on a museum shelf, forgotten by almost everyone except the archivists responsible for keeping track of it. In July of 2025, a research team led by Adeline Morris-Jacobs of Liverpool John Moores University, working alongside Pontus Skoglund's Ancient Genomics Laboratory at the Francis Crick Institute in London, published the findings in the journal Nature under the title "Whole Genome Ancestry of an Old Kingdom Egyptian." The study represented the first time researchers had successfully recovered a complete whole genome sequence from an ancient Egyptian individual, a genuine landmark in a field that had previously managed only partial genetic data, and only from remains thousands of years more recent than this one. The team sequenced the genome to roughly 2-fold coverage, extracting DNA from cementum tissue at the roots of 2 teeth using cleanroom laboratory protocols, is specifically designed to prevent modern contamination. Cementum being a dense, mineralized tissue at the tooth root that has proven, across numerous ancient DNA studies in recent years, to preserve genetic material considerably better than bone or other dental tissue, precisely because its density helps shield DNA from the environmental damage that destroys genetic material elsewhere in a skeleton. The researchers then compared the resulting genetic profile against a reference library of thousands of ancient and modern genomes from populations across Africa, the Middle East, and Europe, allowing them to statistically model exactly which ancient populations best explained the specific genetic signature they had recovered. The individual at the center of the study, now referred to in the scientific literature by the code name Nuwayrat, was radiocarbon dated to between 2855 and 2570 BCE, placing his life squarely within Egypt's Old Kingdom period, the exact era in which Egypt's earliest pyramids were rising and the pharaonic state was still consolidating its power. Prior to this study, the oldest and most complete genetic data available from ancient Egypt came from a landmark 2017 study analyzing 90 mummies from the site of Abusir el-Melek, whose remains dated to a period more than 1,000 years later, spanning the New Kingdom through the Roman era. And even that earlier study had only recovered genome-wide data, rather than a truly complete genome, from a mere 3 individuals. Nuwayrat didn't just add one more data point to that earlier picture. He pushed the genetic record of ancient Egypt back over a millennium in a single discovery, to a period for which no comparable genetic evidence had ever existed at all. To understand why recovering this particular genome required overcoming a genuinely serious scientific obstacle, and why that obstacle had defeated researchers for so long, you need to understand exactly what makes Egypt one of the hardest places on Earth to recover ancient DNA from in the first place. DNA is a genuinely fragile molecule, and its rate of decay after death depends heavily on temperature, humidity, and burial conditions. Cool, dry, stable environments, Arctic permafrost, high-altitude caves, certain types of sealed tombs can preserve usable ancient DNA for tens of thousands of years, which is precisely why so much of the field's most detailed ancient genetic work has come from Siberia, Scandinavia, and similarly cold regions. Egypt's climate represents close to the opposite scenario, persistent high heat, the very quality that helped desiccate and preserve so much organic material in Egyptian tombs for millennia, actively accelerates the chemical breakdown of DNA, degrading it into fragments too short and too damaged for reliable sequencing. Compounding the problem further, the artificial mummification process itself, involving the use of various resins, natron salt, and other chemical treatments designed to halt bodily decomposition, introduces additional contamination and degradation that makes genetic material even harder to recover cleanly. As Harold Ringbauer, a population geneticist at the Max Planck Institute for Evolutionary Anthropology, later explained regarding earlier failed attempts, samples that had been mummified using standard embalming techniques proved to be far poorer sources of usable ancient DNA than researchers had hoped, precisely because the embalming process itself worked against genetic preservation, even as it preserved the visible body remarkably well to the naked eye. This combination of factors meant that for most of the history of ancient DNA research, Egypt remained a genuine blind spot, an almost paradoxical gap in the global picture. Geneticists had built increasingly detailed genetic maps of ancient Europe, the Eurasian steppe, the Americas, and even other parts of Africa, using ancient DNA recovered from burial sites across those regions to trace migrations, population replacements, and admixture events stretching back tens of thousands of years, in some cases producing genomes far older than anything available from Egypt. Egypt, despite being home to one of the best documented ancient civilizations on the planet, arguably the single most intensively studied ancient culture in all of archaeology, remained comparatively dark on that same genetic map, its written records, art, and archaeology extraordinarily rich, but its actual population genetics at the deepest and oldest levels essentially unknown to science. That gap left a specific and long-debated historical question genuinely unresolved. Egyptologists had long known from archaeological and artifactual evidence alone that Egypt maintained significant contact with the broader Fertile Crescent region to its east, The area encompassing Mesopotamia and the Levant, well before and during the rise of the pharaonic state. Domesticated plants and animals, certain administrative practices, symbolic and iconographic parallels, and crucially technologies including the potter's wheel, all show clear signs of having spread from Mesopotamia into Egypt during the 4th and early 3rd millennia BCE in the archaeological period sometimes referred to as the Uruk expansion, named for the great Mesopotamian city whose cultural influence appears to have radiated outward across an enormous area during this formative period. But archaeological evidence of shared objects, technologies, and administrative ideas cannot by itself tell you whether those things spread through trade and cultural exchange between separate, genetically distinct populations who simply borrowed good ideas from their neighbors, or whether actual people, carrying their own DNA, physically migrated from Mesopotamia into Egypt and mixed with the population already living there. For decades, this question could only be argued from indirect evidence, pottery styles, symbolic systems, and administrative parallels, none of which can definitively prove that human beings, rather than just their ideas and their goods, actually made the journey between these two early centers of civilization. Only a genuine ancient genome, recovered from an individual old enough to have lived at or near the actual period when this cultural exchange was taking place, could settle the question directly. And until 2025, no one had ever managed to recover one old enough to actually test it. The genome recovered from Nuwayrat answered the Mesopotamia question with a level of directness that surprised even researchers who had suspected some connection existed. Rather than showing an exclusively local North African genetic profile, as some earlier models of Egyptian population history had assumed, based on the region's deep archaeological continuity, the whole genome analysis found that Nuwayrat's ancestry was best represented by a two-source model. Approximately 78% of his genetic ancestry traced most closely to Neolithic populations of northwest Africa, specifically to genomes recovered from the Middle Neolithic site of Skira Ruwazi in modern Morocco, dated to roughly 4780 to 4230 BCE, representing the deep indigenous North African population base that had inhabited the region for thousands of years before written Egyptian history began. But the remaining approximately 22% of his ancestry traced clearly and specifically to populations from the Eastern Fertile Crescent, showing the closest genetic affinity to Neolithic Mesopotamian populations dating all the way back to between 9,000 and 8,000 BCE. Ancient farming communities from sites including Nemrik and Bonkuklu Tarla in what is now northern Iraq and southeastern Turkey, among the very first places on Earth where humans domesticated crops and settled into permanent agricultural communities, laying the foundation for civilization itself thousands of years before the first Egyptian pyramid was ever conceived. This finding did more than confirm that some genetic connection existed between ancient Egypt and Mesopotamia. It provided, for the first time, direct biological proof that actual people, not merely trade goods and technological ideas, had moved between these two centers of early civilization and interbred with the existing population, generations before this individual was even born. As Pontus Skoglund of the Francis Crick Institute put it following the study's publication, this individual represents the first genetic evidence showing that people moved into Egypt from the Fertile Crescent and mixed with local populations by this early date in Egyptian history, confirming through hard genetic data what archaeologists had only been able to infer indirectly from shared pottery styles and administrative practices for decades, without ever being able to prove it conclusively. Crucially, isotope analysis conducted on Nuwayrat's own teeth, which can reveal information about the geographic region and climate a person grew up in based on chemical signatures absorbed from local food and water during childhood, confirmed that Nuwayrat himself was not a first-generation migrant from Mesopotamia. The isotopic signature was fully consistent with a childhood spent in the hot, dry environment of the Nile Valley itself, eating a typical local diet of wheat, barley, and terrestrial animal protein, with the likely inclusion of Nile fish common to Egyptians of this period. In other words, the Mesopotamian genetic contribution detected in his genome hadn't arrived with him personally. It had already been present in the Egyptian population for some number of generations before his birth, the product of an earlier wave of migration and intermixing that predated him, meaning the actual movement of people from Mesopotamia into Egypt had to have occurred even earlier than his own lifetime, around 2700 BCE. Pushing the true timeline of this ancient migration back further still into a period the genetic record had never previously been able to reach at all, and quite possibly connecting directly to the same broader Uruk period cultural exchange that archaeologists had already identified through pottery and administrative technology, now finally confirmed as involving real human migration rather than cultural borrowing alone. The physical evidence recovered alongside his DNA added a genuinely human dimension to the discovery, transforming an abstract genetic percentage into the outline of an actual person's working life. Bioarchaeologist Joel Irish's detailed skeletal examination found that Nuwayrat 1 stood approximately 160 centimeters tall and died somewhere between the ages of 44 and 64. A genuinely advanced age for the period, when life expectancy was considerably shorter than in the modern world. His skeleton showed pronounced osteoarthritis in his knees, hips, and right foot specifically, along with expanded seat bones and extensive repetitive wear patterns in his arms, a combination of physical markers that Irish concluded were consistent with decades spent working at a potter's wheel, seated for extended periods while performing repetitive hand and arm movements throughout his working life. This detail carries its own quiet irony worth sitting with. The same pottery wheel technology that had itself spread into Egypt from Mesopotamia during the same broader period of cultural contact may well have been the very craft this particular man, carrying Mesopotamian ancestry in his own genome, spent his working life practicing, a coincidence connecting his own body's genetic inheritance to the trade he apparently dedicated his life to. The circumstances of his burial added one final important layer to the picture. Despite the physical evidence of a working life spent in demanding manual labor, Nuwayrat 1 was buried with what researchers interpreted as signs of elevated social status. His body was placed intact, without the resin and chemical treatments of formal mummification, inside a large ceramic vessel, which was then sealed within a rock-cut tomb chamber, rather than a simple pit grave, a level of mortuary care that suggests genuine respect and status, even for a man whose bones tell the story of decades of hard physical work. Researchers noted this combination, clear evidence of manual labor alongside an elevated burial, as a plausible sign of social mobility within Old Kingdom Egyptian society in which a highly skilled craftsman could achieve a level of status and honor in death that his daily physical labor alone might not obviously predict, hinting at an Old Kingdom Egypt where exceptional skill in a trade like pottery could elevate a man's standing well beyond what his manual occupation would typically suggest. And this specific style of burial turned out to be the direct reason his DNA survived at all. Because he died before mummification had become the dominant burial practice in Egypt, his body was never treated with the resins and chemical preservatives that degrade genetic material in later mummified remains. While the sealed ceramic vessel and cool, stable microclimate of the rock-cut chamber protected his remains from the extremes of temperature that destroy DNA in more exposed burials. The very absence of the elaborate mummification process that Egypt would later become famous for is, somewhat ironically, the specific reason this became the oldest successfully sequenced genome from ancient Egypt, rather than one of the countless mummified pharaohs and nobles whose far more famous, far more elaborately preserved remains have so far yielded no comparable genetic data at all. A reminder that the most historically celebrated burials are not always the most scientifically valuable ones. Researchers were also able to compare Nuwayrat's genome against the much later genetic data recovered from the 2017 Abusir El-Melek study, whose 90 mummy sample dated to well over 1,000 years after his death, spanning the New Kingdom through the Roman period. That comparison found only marginal genetic continuity between the two populations, alongside evidence of a further, more substantial infusion of Levantine ancestry entering the Egyptian population sometime between Nuwayrat's era and the later New Kingdom to Roman period, sampled in the 2017 study, with Bronze Age Levantine ancestry accounting for roughly 64% of that later population's genetic makeup, in the best-fitting statistical model. That same 2017 study had also found that modern Egyptians today actually carry somewhat more sub-Saharan African ancestry than either the ancient Abusir El-Melek mummies or Nuwayrat himself, a shift researchers attributed to increased mobility along the Nile, expanded long-distance trade with sub-Saharan Africa, and the trans-Saharan slave trade that intensified over the past 1,300 years. A reminder that Egypt's population genetics kept shifting long after the pharaonic period ended, shaped by later historical forces entirely separate from the ancient Mesopotamian contact Nuwayrat's genome revealed. Together, the two studies, separated by more than a millennium of Egyptian history, begin to sketch a genuinely dynamic picture. Rather than one single founding population that stayed genetically static from the earliest pharaohs through the Roman conquest, ancient Egypt appears to have experienced multiple distinct waves of contact and admixture with populations from the wider Fertile Crescent and beyond, stretching from the Neolithic period detected in Nuwayrat's genome all the way through the later Bronze Age movements reflected in the more recent mummy samples, and continuing still further with the sub-Saharan contributions visible in Egyptians living today. Ancient Egypt, in other words, was never the sealed, unchanging genetic isolate that some earlier, simpler models of the ancient world assumed. It was, from its very earliest recoverable genetic moment, already a crossroads. For more than a century, the skeleton of a man who once spent his working life bent over a potter's wheel sat quietly in a museum in Liverpool, having survived a transatlantic shipment, a world war, and the near-total destruction of the collection housed around it, without anyone realizing exactly what his bones were capable of revealing. It took modern genome sequencing technology, a fortunate accident of burial circumstance that spared his body the chemical preservation that would have destroyed his DNA, and a research team specifically searching for the oldest viable genetic sample Egypt had to offer to finally let a single, ordinary craftsman answer a question that a century of archaeology, pottery analysis, and administrative record-keeping had only ever been able to guess at. Not whether cultural ideas moved between Mesopotamia and Egypt at the dawn of pharaonic civilization, but whether actual human beings did, carrying their ancestry with them into the deep genetic history of one of the world's great civilizations. There's something worth sitting with in the fact that this answer came not from a pharaoh, not from a queen, not from any of the famous mummified rulers whose faces and names we already know from millennia of Egyptian history and countless documentaries, but from an anonymous working man buried without embalming, whose aching joints tell us more about how he actually spent his days than any inscription ever could. The grandest and best preserved monuments of ancient Egypt, ironically, turned out to be genetically silent, their mummification chemistry destroying exactly the evidence modern science needed most. While an unassuming potter's pot, sealed by circumstance rather than design, held the oldest voice ancient Egypt has left us, a voice that, when finally read at the level of the genome, confirmed something written in his bones and blood, long before any hieroglyph was ever carved to record it. Two civilizations that modern history books often treat as separate self-contained stories, Egypt rising along the Nile, Mesopotamia rising between the Tigris and Euphrates, turn out at the genetic level to have already been quietly intertwined by the time Egypt's first pyramids broke the horizon. If a single grave excavated in 1902 and left largely unexamined for over a century could still hold an answer this significant once the right technology finally caught up to it, it raises a genuinely worthwhile question. How many other quiet, ordinary burials, sitting in museum storage rooms around the world right now, completely unremarkable to look at, are waiting for their own moment to rewrite what we think we already know about the deepest chapters of human history? If you found this as fascinating as we did, subscribe to DNA Origins for more documentaries uncovering the genetic history hidden inside the world's peoples. Next week, we're returning to West Africa to trace what modern genetic studies have revealed about the deep, surprising origins of the Yoruba people. That video is already on the channel.